Flotation technique, its use and its importance. So what is a flotation technique? It is a simple test tube flotation technique used for routine veterinary tests, specifically to diagnose or detect nematode and cestode eggs, indicating mature parasites live inside the animal. This method is useful to use in preliminary surveys to establish which parasite groups are present in an area. There are 4 samples collected from Minapan, Tulunan from the southernmost part of Cotabato Province. The sample 1 was collected on the 16th of October 23 at 7:30 p.m. from a 2-month-old male pig. For samples 2, 3, and 4, they were collected from the same source or same pen, which was said for the pigs to be fed with grower SPM3. These pigs were raised in the backyard or an outdoor house. Preparation of saline solution. You may need a weighing scale, 800 grams of rock salt, a beaker, 2 liters of distilled water, and a stirring rod. For the procedure, you have to weigh 800 grams of rock salt, pour the rock salt into a beaker, then you add 2 liters of distilled water, and mix thoroughly until the salts dissolve completely. And now moving on to the flotation technique, you may need the following materials. We have the gloves, test tube rack, weighing scale, beaker, conical flask, tongue depressors, fine mesh sieve, glass slide, cover slip, petri dish, stereo microscope, syringe, graduated cylinder, pipette, and a container with the sample feces. For flotation technique, 3 grams of feces will be weighed and placed in a plastic cup. The feces will be broken down into pieces and then added with 50 ml saturated saline solution. This will be mixed vigorously using a stirring rod. The fecal mixture will be sieved to remove the debris until a homogeneous suspension will be seen. The test tube will be filled with the suspension until a convex meniscus at the top of the tube will be created. A cover slip will be placed on top of the test tube, ensuring no fluid will outflow from the tube so that the eggs will not be poured out. The test tube will be left to stand for 20 minutes and the cover slip will be lifted off together with the drop of fluid adhering to it. The cover slip will be placed onto a glass slide and examined immediately under a microscope. So let's have an overview of common intestinal nematodes of pigs. Here we have the Eimeria species, Ascaris suum, Strongyl type, Trichuris suis, Strongyloides ransomi, and the Isospora suis. For Eimeria species, this genus of coccidian is characterized by being monoxenous, mainly infecting the gastrointestinal tract. It is characterized by having oocysts with 4 sporocysts, and when sporulated, each sporocyst contains 2 sporozoites. The wall of the oocyst is often brownish in color, and many species have a polar cap. The members of this genus all have direct life cycles, thus the general form of the direct coccidian life cycle is represented by the genus Eimeria, species of which include gastrointestinal parasites of a wide range of vertebrate hosts, and that includes the swine. For Ascaris suum, it is a ubiquitous and pathogenic parasite of swine, and the adult worms are about 30 cm long and white to cream colored, with 3 large lips typical of the ascaroids. For the ascarid eggs, they are relatively thick-walled, containing a single cell when passed in the feces, and are usually sufficiently distinctive to permit identification of the species. Strongyl type of eggs are from the superfamilies Strongyloideae, Trichostrongyloideae, and the Encyclostomatidae, which have thin-walled ellipsoidal eggs containing an embryo in the morula stage of development, which is the same stage found in the host feces. Metastrongyloideae are also thin-walled and ellipsoidal, but the development stage is deposited in the host tissue. For Trichuris suis, it is a whipworm and have an oval egg, which is about 60 by 25 micrometers, and have a yellow-brown color with a bipolar plug, which is a lemon shape in appearance. For Strongyloides ransomi, the eggs are thin-shelled, larvated, and are 45 to 55 by 26 to 35 micrometers in size. For Isospora suis, the oocysts are ellipsoidal to subspherical, which measure up to approximately 20 by 15 micrometers, and have a thin, smooth shell. Upon performing the flotation technique, 4 out of 4 samples are found to be positive with Eimeria and Strongyl types of nematodes. There is an increased prevalence of gastrointestinal parasites, which may result from the accumulation of feces for extended periods, usually more than 3 to 4 days, allowing the eggs to mature in favorable moist environments that promote egg survival. Understanding the parasite's life cycle is crucial for prevention, such as implementing measures to interrupt this cycle and prevent reinfection, and alongside using antihelminthic treatments is very essential.